Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 7th January 2021, as we are into the quick revision notes for all the chapters after having completed the syllabus, so that this notes will be handy during your examinations. You will have a quick revision of each chapter. So as part of it, we are dealing now, after having done chapter 19, we are going to chapter 20 and locomotion and movement. And for your memory, I want you to recollect that this chapter has been reduced. This has got only muscles and skeletal structure. That's all. Other things are deleted for you. So it has got limited syllabus in this chapter. So let's go for the definition. What is locomotion? It is the voluntary movement of an individual from one place to another. So walking, running, climbing, swimming are examples of locomotion. Even you must have seen that the animals show locomotion, whereas plants, leaving this hydrophytes, land plants, they don't show locomotion, but they show movement. That is the swing of the branches of the trees. So you must have that clear-cut difference between locomotion and movement. Locomotion is displacement of area. Movement is, the area is same. The organism will not move, but it will have a swing of the arms, swing of the parts. So all locomotions are movements, but all movements are not locomotion. That's why I said. So types of movement. Cells of the human body show three main types of movements. What are those? Amoeboid movement. You must have seen how amoeba moves underwater. Under microscope, you must have noticed. Ciliary, of course, you have seen paramecium with a lot of cilia, hair-like structures. Then, of course, your own muscles, muscular. So three types of movements are possible. So, macrophages, macro, big ones, leukocytes in blood exhibit amoeboid movements. Okay, 360 degrees movement will be there. Macrophages and leukocytes, a type of WBC. Coordinated movement of cilia in trachea, respiratory system, to remove dust particles, and passage of ova through fallopian tube is the example of ciliary movements. You need to remember these examples in the examination. These examples you need to remember. Movement of limbs, jaw, tongue need muscular movement. Contractile property of muscles is used in movement in higher organisms, including us, human beings. So this is the contractile property. So muscles are specialized tissues of mesodermal origin. You know, there are three layers in the embryonic stage. That is ectoderm, outside, mesoderm, middle, endoderm, inner. So muscles are all taking their origin from the mesoderm. They have property like excitability, contractility, extensibility, and of course elasticity. All this we experience whenever we make our muscles to move. So based on their location, there are three types of muscles. As all of us know, skeletal muscles, the muscles that are attached to the bones, visceral muscles, inner ones, cardiac muscles, special muscles that are present in the heart. So the skeletal muscles are associated with the skeletal system, bones, alternate light and dark bands, that is striations will be there. They are voluntary and locomotory and change in body posture function. They are responsible for our shape. And visceral form inner wall of internal visceral organs, non-striated, smooth muscles. Involuntary muscles, involuntary, not voluntary, assist in movement of food through digestive tract and movement of gametes. On the other hand, we said cardiac, they are special muscles, they are present only in the heart. Cardium means heart, cardiac muscles, having branching pattern, alternate light and dark bands, involuntary in action. These are blend of smooth muscles and rough muscles. and they are involuntary. Thankfully they are involuntary. Okay, otherwise you say that, yeah, let my heart be stopped, it won't stop, no. And when it stops, let my heart function again, it won't start functioning, no. So that is the reason we are saying involuntary. Skeletal muscle is made up of muscle bundles, these are called fascicles, held together by collagenous connective tissue called fascia. How a muscle, when we take the section of this is how the skeletal muscle section will appear. Now what is that you need to observe? First, fascicles, muscle bundles, you can see the bundles, and concentrate or focus on one bundle. You will see inside the bundle muscle fibers, okay, the myoplasma we say, and then sarcolemma, the outer membrane of the muscle, sarcolemma, and lot of blood capillaries which supply blood to the muscles rich. So the skeletal muscles are having these parts. Each muscle bundle contains number of muscle fibers. Each muscle fiber is lined by plasma membrane called sarcolemma. You must remember this. Enclosing sarcoplasm, just like cytoplasm. Sarcoplasm, liquid present in sarco. Partially arranged myofibrils are present in muscle bundle having alternate light and dark bands due to the presence of a protein. In fact, two proteins, one is actin, another is myosin. These are very, very important proteins. They have to be present in the muscle. And if there is any gutbad, this actin and myosin not there, then the muscle starts degenerating. Remember that one disease I told you, myasthenia gravis, related to this myosin. So light bands contain actin and is called I band. Why I band? Isotropic band. And the dark band contains, as I said just now, myosin, called A band, and isotropic band. Both strands are present parallel to each other in longitudinal fashion. Recollect the diagrams that you have drawn. In center of each I band is elastic fiber called Z line. In the middle of A band, that is an isotropic band, is the thin fibrous M line. The portion of myofibrils between two successive Z lines is the functional unit of contraction called sarcomere. At resting stage, thin filament overlaps the thick filament. This part of thick filament not overlapped is called H zone. So for all this, how to identify? This is the way, draw the diagram. You can see that I band, H band, okay, Z zone, all these you can see here. And overlapping of the myofibrils, all this. So all this is possible because of the protein and structure of contractile protein. Each thin filament, that is actin, is made up of two F actins helically wounded to each other. Two filaments of another protein called tropomyosin runs close to it. Look at that, tropomyosin is running close to it, and this is F actin and troponin. And this is how the protein, contractile protein structure will be there. A complex protein, troponin, is distributed at regular intervals on the tropomyosin, as you are seeing here. Each myosin filament is made up of many monometric proteins called meromyosins. Each meromyosin has globular head with a short arm and tails. Globular head has ATP binding sites. That's how they are energy rich. And how muscles contract, what is the mechanism involved? And I have told you when I was dealing in the regular class that Ca2 plus is very, very important. And remember that calcium and potassium and sodium potassium pump I said is very, very important for normal functioning of heart. Similarly, calcium is required not only for bones, even for the muscles. In what way this calcium is important? Let us see. So mechanism of muscle contraction, normally this is a 3-mark question, so you need to draw diagram also when you are along with the explanation. So the mechanism of muscle contraction is explained by sliding mechanism theory. Remember that? Sliding means slipping one over the other, in which the filament slide over thick filament. Thin filament slide over thick filament. Muscle contraction start with the signal sent by central nervous system via motor neurons. Neural signal releases a chemical called neurotransmitter. For example, acetylcholine is a neurotransmitter to generate action potential in the sarcolemma. This causes the release of Ca2 plus from sarcoplasmic reticulum. Ca2 plus activates actin, which binds to the myosin head to form a cross bridge. These cross bridges pull the actin filaments, causing them to slide over the myosin and thereby causing contraction. So Ca2 plus are then returned to sarcoplasmic reticulum, which inactivate the actin. This is cross bridges are broken and the muscles relaxed. Now you see here, this is actin filament, this is myosin filament. You can see the bridge with the involvement of ATP, the expenditure, and you can see the formation of myosin head here. Formation of cross bridge has taken place here. This is the cross bridge, and now the sliding is taking place. One is skipping of other, and then the cross bridge is broken here and the muscle gets relaxed. So how many types muscles can be classified? The red fibers, aerobic muscles, contain myoglobin that has plenty of mitochondria to use large amount of oxygen stored in them. Red meat, we say. White fibers, on the other hand, the muscle fibers containing less number of myoglobin are called white fibers. So in our next session, we will see the bone system, skeletal system. So you need to practice many diagrams, draw this mechanism of particularly muscle contraction is very, very important. It's a 3-mark question. So keep on practicing.